Thank you to Gatsby for the fantastic opportunity to come and join and share in this amazing summer school. And I hope all of you have been really enjoying the scientific discussion so far this week. And now we're going to focus this morning on disease. So my lab focuses mostly on plant disease. And we're going to be looking this morning at how we can safeguard one of our oldest and most precious food crops, wheat, from these cereal fungal invaders. And so today we're going to be looking at these cereal killers, these wheat rust, and they're also known as the polio of agriculture. And this is because of the sheer devastation that they cause to wheat production every single year. And then we're going to be looking at how we can use genomic-based approaches to try to track individual strains of these wheat rust fungi. And we're going to consider the development of one technique that we use in the lab called field pathogenomics. And then we'll go on to look at how we can also use these exciting sequencing technologies and the power of genomics to uncover new sources of host resistance too. So looking on the wheat side of this at how we can actually better improve the resilience of our crops. And then finally, we're going to be considering this morning how we can also work hard together to enhance accessibility to these fantastic technologies and also genomic-based approaches to improve pathogen surveillance and diagnostics for everyone worldwide by improving accessibility. And here we're going to consider one of the programs that we run in my lab called MARPLE Diagnostics, which stands for Mobile and Real-Time Plant Disease Diagnostics. So now to introduce you to the wheat rust fungi, which are also known, as I mentioned, as cereal killers. So these wheat rust diseases are caused by 3 different fungal organisms that cause 3 distinct diseases of wheat, known as leaf rust, stem rust, and yellow rust. And collectively, these 3 wheat rust fungi cause a huge amount of damage to wheat production every single year. So if we consider them collectively and we look on a global scale now at the damage that they cause to wheat production, actually every year on a global scale, they destroy 15 million tons of wheat. And so this equates to an annual loss of over 2.9 billion US dollars. So this sounds like a really, really big number. And indeed, this is quite a big number. So if we think about this number and we think about the amount of wheat that is destroyed by these cereal killers every year, and then we take that wheat, and of course wheat can be used for many different purposes, but if we take that wheat that's destroyed and we just use it to bake bread, it's enough wheat to bake over 25 billion loaves of bread. And that's enough bread to satisfy the annual consumption of over 425 million people, or indeed over half the population of Europe. And so this is a huge amount of wheat that's destroyed by these cereal killers. And as we look to the near future, just over the next 10 years, it's thought that wheat consumption is also going to increase by around 11% over that time period. And so you can see that if we can actually curtail the trail of destruction caused by these cereal killers, that can really then start to close that missing production gap and ensure that we can also safely feed our ever-expanding global population. But the fact that these cereal killers are actually fungal organisms is, in human history, quite a new discovery. And if we want to really understand kind of the history of plant disease and how our understanding of the history of plant disease has really changed over time, we have to turn our attention now back to our ancient counterparts and their reliance on the supernatural religion, natural philosophy that really underpinned their understanding of plant disease for so long. And into modern times. So if we go back now in time and we look at 700 BC, this was the period where we had the rust gods. And so in Greek, this was the Apollo of rust, and in ancient Rome, this was the rust god Robigus. And these were considered supernatural plant pathologists. And so they were worshipped every year in order to protect crops from these wheat rust cereal killers. And in the case of ancient Rome, Robigus also had their own festival known as the Robigalia Festival. And every year on the 25th of April, people would come out and they would take red-haired animals and they would sacrifice them to the rust god Robigus, hoping that this would then protect their crops from these wheat rust cereal killers and ensure a bountiful harvest. But of course, sadly, this was not very successful. Sacrificing red-haired animals really didn't help with plant disease protection. And then, of course, the scale of devastation caused by these wheat rust pathogens also brought the attention of many, many ancient philosophers, including, of course, Aristotle. And Aristotle was one of the first philosophers to articulate the theory around spontaneous generation. And so in spontaneous generation, he believed that actually living beings were created by non-living beings. And so somehow these non-living beings could magically create living beings. And in the case of the wheat rust, he thought that what he saw out there in a farmer's field was created by warm vapors. Okay, so somehow these magical warm vapors were turning into what he saw being damaged in their wheat fields. And if we look then over the next few centuries, our understanding of plant disease really changed very, very slowly. And it wasn't until the 1700s that we have the scientific name then attributed to the wheat rust. And this was where it was then called Puccinia. And so this is the rust genus, Puccinia. But even at this time, when we have the scientific name attributed to the wheat rust, actually at this point, Puccinia, if you go back to this description, is described as a plant. And so they were described it as a plant, and they also thought that this plant came after you saw the disease in the field. And so they didn't think it was the causal agent at this point. It wasn't until the phenomenal work of Hendrik Antoine de Barry that finally he was able to show that fungi could cause plant disease. And so this was in the mid-1800s. But also the phenomenal work of Hendrik Antoine de Barry also went on to resolve the very complex infection cycle of these deadly serial killers. And now to really appreciate the work of Hendrik Antoine de Barry, we're going to have a look at that very complicated life cycle of these wheat rust fungi. And so here, this life cycle is extremely complex. And so these fungal organisms create 5 different types of spores, and they infect 2 different plants that are completely unrelated. Here we have the asexual part of the life cycle where the fungus is infecting the wheat plants. And so it's creating what we call uridinospores. And those uridinospores are clones of each other. So it's through asexual reproduction that it then goes on and infects and spreads the disease from one wheat plant to the next. But at the end of the growing season, this fungus changes. And this is the case for all 3 of these wheat rust fungi. They follow broadly the same infection cycles. And at the end of the growing season, when our farmers go out and they harvest our crops, actually the fungus changes and it goes into a different state, and it creates what we call teleospores. And these are black, dark teleospores that are very hardy. And in the case of wheat stem rust, it can't survive the cold winter months in its uridinospore stage. It has to go on and create teleospores in order to survive cold winter months that we have, for instance, here in the UK. So in any temporal climates, you will see it has to go through this process. For leaf rust and also yellow rust, it can survive in this other state. But for stem rust, it has to go down this path in cold climates. And so these teleospores are created, and this is kind of a dormant stage of the fungus. And so it creates these hardy, overwintering teleospores, and at this point, the fungus goes to sleep. And so the fungus then will sleep throughout the winter months, and only in the spring will it reawaken when it can infect its alternate host. So it will then create what we call basidiospores, and they go on to infect a totally unrelated plant known as barberry, which includes many species in the Berberis genus. And then at that point, the fungus undergoes sexual reproduction. And we're all familiar with sexual reproduction. This creates new genetic variants at this point, and so these new genetic variants might have new abilities. So for instance, wheat doesn't just come in one flavor. We have lots of different varieties of wheat, and some of them might be resistant to infection. Some of those varieties might be susceptible to infection. And at this point, when it goes through sexual reproduction, it might create new variants, new strains that are able to overcome some of the varieties that were previously resistant, because these new strains have new abilities. And so those new strains are then in the form of aciospores. So this is the type of spore that is representing the sexual reproduction stage. And then these aciospores can go on and they will spread the disease back into the wheat crop. And so aciospores are created in these tube-like structures that form on the underside of barberry leaves, this hedgerow shrub. And then they are released in large quantities on the underside of these leaves. And these spores don't travel very far, so it's thought that they will spread the disease from a hedgerow that's next to a cereal crop back into that cereal crop or wheat field. And so it will spread the disease, completing the whole infection cycle. So now we've gone from wheat through barberry and back again to wheat. And so this is the way that we get these new genetic variants being created. But of course, it's the part that occurs on wheat that tends to get a lot of attention. That's the part of the life cycle that most people spend their time considering. And indeed, this has taken up a lot of the work that people do around the world studying these wheat rust fungi. They mainly focus their attention on the asexual part of the life cycle. And for today, we're going to focus our attention on this part of the life cycle where the fungi are infecting wheat plants. And so this is where they create these stunningly beautiful asexual uridinospores. And these are released from a leaf or from the stem of the plant in pustules. And these pustules are where the fungus has ripped open the epidermis, releasing these spores. And these spores then blow on the wind, so they move the disease from one plant to the next, from one field to the next. But they can also blow very long distance, so they will move the disease potentially from one country to the next, or even between continents. So they can be caught on wind currents, and that can really help to move the disease around the globe. But also, of course, we don't have just the potential for this disease to naturally blow on the wind across the globe. We can also help to move them around the planet, and this is because these spores are very small. They can easily get caught on your clothing, and you can help to move them around the globe. And to illustrate this point, here we have a diagram which shows the number of people that are moving using air transport in just one day. And so you can see that there's connections all over the globe here. And of course, if you capture some plant pathogens on your clothing and then move from one location to the next, you can easily spread that disease to a new territory. And so as people continue to vacation in lots of exciting, exotic locations, this of course then helps to move plant pathogens from one location to the next. And so now we can't just rely on predicting the movement of these plant pathogens using wind movement. We also have to consider people movement too, and also, of course, trade. And so plant pathogen movement is becoming increasingly unpredictable and difficult for us to track. But we also have to keep in mind that we have these new strains that are constantly arising, and we have to keep track of where they are around the globe due to the fact that they might infect different wheat varieties. So to illustrate this point, here we have one particular strain that is present in this particular area, and we have two different flavors of wheat. We have two different varieties of wheat. The top one here is susceptible to this particular strain, so it is diseased. The bottom one here is resistant to this particular strain, so it's nice, happy, and healthy. We're really happy at this point. This plant is looking happy and healthy. But unfortunately, a new strain can easily move into an area, blowing on the wind, or we can facilitate this process. And then you get a new strain moving in that is now able to infect both of these wheat varieties. And so now the one that was happy and healthy has succumbed to disease, and it's classified as being susceptible. And so new strains moving into an area can be really worrying and something that we have to keep on top of. And to consider this further, we can take an example from Europe, and we can look at yellow rust. So yellow rust, one of our serial killers, in 2011, it underwent a bit of a shift in Europe. And at that point, we had the arrival of new strains of yellow rust. So in 2011, we got new strains of yellow rust moving into Europe. And we can see this in this diagram here. If you have good eyesight, you'll be able to see it along the bottom. We have the years. And all of these bars, the colors of these bars, represent different strains. And in 2011, you see this pink area appears, and then this pink area really takes over. So this is the arrival of a new strain, and then it's becoming the most dominant strain that's present in Europe. And so this was the arrival of new strains of yellow rust moving into Europe in 2011. And we know now that these new strains came from the Himalaya region. We don't know how they got here, but they moved somehow into Europe. And what was concerning about seeing new strains in this region was the fact that these new strains could actually infect new wheat varieties. So wheat varieties that were previously resistant could now be infected by these new strains. And so this was very concerning at this point. We then had to work with our breeders to try to screen our material and make sure that we also then could get some wheat varieties that were also resistant to these strains and fast track them for release. And so you can see the kind of scale of devastation that could be caused by new strains and how rapidly they spread potentially within a pathogen population. And so then how do we actually use genomics to keep track of these new strains? So how can we use genomic-based approaches to keep track of these new strains that might be arriving or being created within our countries? And also use genomic-based approaches and all these exciting sequencing technologies to enhance our resilience to these ever-evolving threats. And so here I wanted to introduce you to a technique which we call field pathogenomics, which is a genomics-based technique for gaining insight from both the pathogen and the host to inform disease management. And so we're going to go through this very slowly on how this technique actually works. But the basics of this technique is we take an infected leaf, like you see here, or a stem of a wheat plant, and then we use the power of genomics, and we sequence this leaf in order to know which strain is present and which wheat variety is present. And then we can analyze that data. So to look at this in a bit more detail, what we do is we go out and we collect samples from the field. These will be infected leaf samples, stem samples, or we work with collaborators to get samples from all over the globe. And then we do what we call transcriptome sequencing. And so we extract RNA from these samples, do transcriptome sequencing, and magical data analysis in order to type our fungal strains. So how do we actually process our samples for transcriptome sequencing? Well, when we do transcriptome sequencing, what we're doing is we're sequencing all the genes that are present of both the fungus and the host plant, so our wheat plant, that are turned on during that interaction. So we're capturing all the genes that are turned on during that interaction. And we take our pieces of leaf, in this case, and we extract mRNA from those samples. And then what we do is we fragment that RNA into smaller pieces. We then convert that RNA into complementary DNA, and then we take that cDNA that we've created, and we have to then process that cDNA. So we take that cDNA that we've made from our RNA, we've extracted from these samples, and we process it. And the reason we have to then process this cDNA is we have to add something to this cDNA in order to do sequencing. And so what we add are what are called adapters. And so we add these special little nucleotide sequences to the end of all our cDNA fragments, and we do this for two different purposes. So once we've got our cDNA, we will add adapters, which are small little nucleotide sequences, which tell the machine, the sequencing machine, that this is a piece of cDNA. So it helps the machine to recognize this cDNA molecule. Also, at the same time, we add another sequence of nucleotides, and this particular sequence is unique to each individual sample, and it's what we call a barcode. And so we add that barcode to the samples too, because that allows us, when we do our magical data analysis, to make sure that all the data from one sample can be grouped together. So we can identify which bits of data came from which samples using those barcodes. And so once we've processed our cDNA, we then do the sequencing process. And for most of these projects where we're doing transcriptome sequencing, we will use these very large Illumina sequencing machines, such as the one shown here, which is present. This is a picture taken from the Earlham Institute in Norwich. And we will use these large immobile sequencing machines because they're really the workhorse of the lab. They're very cheap to run, and we can generate a lot of data from each of our samples. And so we create really vast data sets, such as one shown here, from each of our samples. And each of these data sets, again, it captures all the genes that are turned on in both the pathogen and the plant. And these data sets can be several gigs large. So these are really large data sets that we want to process. And so what do we gain from doing our field pathogenomics technique? So what information can we get from doing transcriptome sequencing? Well, we can gain actually two different pieces of information. So we can type our strains, so we can do genotyping. And to explain how we do this, we take all of our samples and we take the data that's been created. And so this is all the genes, in this case, of the pathogen, and also all the genes of the plant that are turned on. And we will separate them and we will analyze them separately so that we can analyze the particular strain that's present of the pathogen, and we can look at the sequence for the plants too and type the individual varieties. And so we do that by lining up all the sequences, as you can see here, and then looking for similarities and differences in the sequences. And any differences, like you can see here, we have a T and a T in these two samples, strains C and D, and we have a G and a G for strains A and B. So these single nucleotide differences are known as single nucleotide polymorphisms. And so these are the differences in sequence between individual strains. And we can use those sequences to create what we call a phylogenetic tree. And you can think of a phylogenetic tree like a family tree. And so when we think about this tree, all of these branches that connect these individual strains that we draw are actually related to the number of differences we see between our individual samples. So if we think in the context of a family tree, if we run through this connection, it's quite short. This line that we've drawn between A and B is quite short. And so they'd be like brothers and sisters. They don't have very many differences between them. But if we take strain A and then we compare it to strain C, you can see if we follow this line, it's much, much longer. So these might be different distant cousins, for instance. And so these line lengths represent the number of changes that we see between samples. So it can help us to define whether we have a new strain in an area. So for instance, if we always type our strains in our sample as being A and B, and then suddenly we see C, then we know that something has happened. We've got a new strain in that sample. And so by doing transcriptome sequencing, we can type the precise strain that's present in a sample. And also at the same time, we can use a similar process to type the wheat variety that's present in that sample. But also, because we're doing transcriptome sequencing and we're looking at all the genes that are turned on in both the pathogen and the plant, we can do something else. And we can look at the expression of every gene that's also captured during this process. And so genes in this particular example are shown by these little arrows. And we have gene 1 here, and we have some transcripts being associated with that gene. So this gene would be classified as on. We've got data that relates to it. This gene is on. The second gene is off. We don't have any data relating to that. And the third gene is on again. But also you can see that the amounts of data, as we've represented with these squiggly lines, is much higher for this particular gene versus this gene. So this gene is actually turned on more than this gene here. And so we can also get information about gene expression using transcriptome sequencing, which is very, very valuable. And we're going to look at the value of this data over the next few slides. And so using this field pathogenomics method, you can see that using this genomics-based approach is very, very powerful. We can gain lots and lots of information. We can gain information for the pathogen, so looking at our rust fungi, we can work out which particular strain we have in our sample, and then we can also look on a broader scale at where those strains are present around the world. We can track them over time, and we can see how they might be moving. But also, at the same time, we get information on the plant as well. And we want to focus today more on the plant side of this interaction through this lecture. And so because we're sequencing an infected leaf, we get the kind of intimate association between the plant and the pathogen when we do our sequencing. And so here you can see this is one of our rust fungi that's got a spore that's landed on the leaf surface. It's then germinated, it's then entered into the underlying mesophyll tissue, and it's proliferated. Eventually, after a couple of weeks, it will then burst through the surface with the production of pustules with all of those individual uridinospores again, which will get blown on the wind to infect a new plant. And so when we do our transcriptome sequencing, we're capturing this entire moment when we're doing transcriptome sequencing. And so we get that intimate connection between the plant and the pathogen, and we can understand more about how the plant is responding to being infected. And so now I wanted to show through an example how we can actually utilize the differences between the response that plants make between a susceptible interaction and a resistant interaction in order to find new sources of resistance. And so you can imagine that when our fungus infects our plant, and it's very, very successful, at this point these fungi, they will completely reprogram this plant. And so to be successful, the fungus infects this plant and it has to gain everything it needs to survive from this plant. So it will derive all the nutrients it needs from that plant. And also, when you infect this plant, the plant's not very happy about this process. So the plant will try to fight back. And of course, trying to fight back is not a good thing for the pathogen. So the fungus doesn't like the fact that the plant's trying to fight back. So it will also turn off all those processes. So the fungus has to actively turn off the processes that defend the plant from being attacked. And so this fungus is actually reprogramming this plant for its benefit. It's making sure that if it needs particular nutrients, it's turning on those processes. It's making the plant make more of them, so that then it can get what it needs to survive and prosper. And it's also turning off all the defense processes. And that's why you have this successful pathogen and why this plant is so diseased. But then you can think, well, what happens if we actually knew what the fungus is doing to this plant? What happens if we understood which were the parts that it needs to change in this plant? And what happens if we could understand those elements and we could work out what those elements are and we remove them? So if we were to remove all of the key elements that the fungus needs from this plant, and we were to remove them, delete them, then we could actually take this plant and turn it into a healthy plant, because we've taken away something that's absolutely essential for the fungus to survive. And of course, many of those components might also be needed by the plant to survive, so we have to be careful about what we remove. But we can actually take away single elements from this plant, so single genes, that could then potentially turn this plant from being susceptible to being resistant, because we've removed something that the fungus absolutely needs to survive. And so that's what we call susceptibility genes, and we're going to now look at how we can identify those elements that are critical for the fungus to survive. And we can do that by utilizing our transcriptome data. So we know that disease and how it progresses is different when you have a susceptible interaction and a resistant interaction. So as we've seen in the case of a susceptible interaction where the fungus wins and the fungus is very happy and the plant's not so happy, in this case the fungus has reprogrammed this plant to survive by itself and the plant's not very happy. But in the case of a resistant interaction, well, the plant won, so the plant wasn't reprogrammed. And so we can actually then compare these two to work out what is the elements that are being reprogrammed here to allow the fungus to survive. And so we know that disease progression is different in a susceptible and a resistant interaction, and we want to know what's causing that difference. So what are the elements that are only switched on here because the fungus has actively started to turn these on? And the way we can do that is through our transcriptome sequencing, looking at gene expression, understanding which genes are turned on and off in both of these situations. And so we know from our example earlier that some genes are active. So here we have our example again of our 3 genes. This one is on, this one is off, and this one is on, because we have transcripts in our data sets that relate to these 2 genes, but we don't have anything relating to this particular gene. So we know this one is on. And we know that we see differences between our susceptible plant and our resistant plant in this case. And we also know that the level of expression varies. So some genes will be turned on a small amount, some genes will be turned on a lot. So we can also look at the amount of data that's relating to each of these genes, the number of transcripts that are being created, and that allows us to work out the amount of expression for each of our genes. And so then we can compare the two. We have all of this valuable information from our transcriptome sequencing, and we can compare the two. We can look at which genes might be only turned on here because they're being actively reprogrammed by the fungus. And we do that by transcriptome sequencing and then linking the expression profiles to our particular interactions. So we're looking for things which are specifically being reprogrammed and turned on here in our plant. And then once we've got a theory from our data, of course, we have to actually test this and see whether or not that element is required for the fungus to cause disease. And the way we do this is we identify mutant plants. So we can find mutant plants, and then we can disrupt our single gene that we may have pulled out and thought this might be the critical element that the fungus needs. Well, we can make mutants in order to work out whether our theory is correct. So ultimately, we're trying to find genes that we can disrupt the function of by working out which elements are required by the fungus to cause disease. And if we disrupt their function, we could potentially get a resistant plant, and that would be very valuable for plant breeding to improve disease resistance. And so to illustrate this a bit further, we're going to take a look at a real example from the lab. And so here we have a gene that encodes a branched chain amino acid aminotransferase, and you don't really need to know exactly what it's encoding, but you can see here we have very high expression. We have a lot of transcripts in a susceptible interaction, shown in orange, against very few in a resistant interaction. And so maybe this is being reprogrammed by the fungus, and it's being switched on by the fungus in this diseased plant. And so then we can find mutants. And so here we're very lucky. At the John Innes Center, we have on site, we have a EMS, so a chemical set of mutants that we can call upon. And these are in what we call chronos. So this is a particular variety, particular flavor of wheat, where we have two different genomes. And so we have an A genome where we have 2 copies of the gene that we're interested in studying, and a B genome that also has 2 copies of this gene that we're interested in studying. And so we need to remove the function of this gene in both the A and the B genome. So we find mutants that do that for us. Then we cross them together in order to create a plant that has no functional copies of this gene. Okay, so we thought this is a critical element that the fungus might need to live and prosper on this plant. So we've removed this element, and then we want to work out, is this actually true? Can the fungus survive without this element being present? And so now we've got mutants, we can actually test that theory. And here we have our wild type plant. So this is just a normal plant which still has that element present. And you can see we have lots of these individual pustules on the leaf surface, and that is signatures of infection. And so we have lots and lots of infection on the leaf surface. The gene is present in this plant, and it's very, very susceptible to infection. So now we're going to look at our mutant plant, the one where we've removed the one gene that we think could be one of the many that might be critical for the fungus to survive. And we remove that gene, we infect those plants, and we don't see any infection. Okay, so now we know that that gene is actually critical to supporting the fungus to survive and prosper on these plants. And so this is a really great discovery. We've now found a gene that is required by the fungus to cause disease. And so if we remove that gene, we can then potentially make resistant plants. And so then at the John Innes Center, what we do when we make these great discoveries is we progress these discoveries into the hands of the plant breeding companies, who will then take those initial discoveries and start to progress them into real-life wheat varieties that our farmers in the future will be able to grow. So then they'll test them and see whether or not they're viable as an option to use in real life in plant breeding. And we hope that by providing a number of these genes where we can see that they are required by the fungus to cause disease, these will act as new sources of resistance that we can use in disease management. So we're now progressing those into the hands of the breeding companies. And so you can see from all of this work that this field pathogenomics technique that we created is extremely valuable. We're gaining so much information, both by typing individual strains of the fungus, working out how they're moving around the globe, and also on the host side of this interaction. But one of the real downsides of using transcriptome sequencing we saw earlier, which is the really big Illumina sequencing machines, so the really large sequencing machines. Not everybody has access to them, so that really prevents people from utilizing these machines. And so we wondered whether or not we could take the essence of what we created in the generation of the field pathogenomics technique and take these genomic-based innovations and make them into something that's more portable and accessible to everybody, irrespective of access to resources. And the work I now wanted to talk about specifically focuses on disease diagnostics, so looking at typing individual strains of the fungus. And we wondered if we could take our field pathogenomics method, take out the part which is around disease diagnostics, so around typing individual strains, and see whether or not we could convert this into something that could actually be utilized in resource poor regions, so improving access across the whole landscape, across the entire globe. And this would then enhance disease diagnostic capacity in these areas. And so why are we interested to do this? Well, sadly, unfortunately, in these kind of resource poor regions, this is often where kind of disease epidemics are really hard felt. And so, for instance, if we take this example from Ethiopia back in 2010, which is a country where we work. So in 2010, this is in recent history, one of the largest wheat yellow rust outbreaks that they saw. In this particular year, over 600,000 hectares of crops were affected. That's over a third of the country's wheat production area. The losses are equated to over $250 million US. And so this was a catastrophe for Ethiopia. They lost a huge amount of production in that particular year. And of course, in Ethiopia, this had wide-reaching implications for the people living in the country. It really was a catastrophe, this outbreak of yellow rust. And so, unfortunately, from this situation, of course, this was a very devastating situation in Ethiopia. But from this situation, the whole community came together to try to see what we could do to prevent this from happening in the future, to try to protect people in Ethiopia from these really big disease outbreaks. And what the community did, working with Ethiopian scientists, was to create what we call a wheat rust early warning system. So this was created by an entire community effort. And this consists of a number of different components to try to prevent that kind of high level of epidemic that could ever happen in the future again from ever happening. And so this was a monumental effort. And I'm just going to introduce you to a couple of the components that are contained within this early warning system. So they have, for instance, surveys. So they have a little mobile phone app, so they get alerted if there's disease in their area. So farmers can then take action if they get these alerts on their mobile phone. They also have invested in training people in country to make sure that they can also do work in country on the rust strains that they have present. And so they will then be able to go to the field, take the strains from the field, and they'll be able to grow them in the lab, and they'll be able to then work out which wheat varieties are vulnerable to the strains they currently have in country. And they also now have these really large screening nurseries where they're able to screen all the different flavors of wheat, so all the different varieties that they grow in Ethiopia. They're able to screen them with the current spores that are floating around in the country, and they're able to work out which varieties might be resistant, and then they fast track those into the hands of the farmers. So they make sure any resistant varieties get into farmers' hands as quickly as possible. And then also at Cambridge University, led by Chris Gilligan and the Met Office, they've worked together to also implement disease forecasting models. And so here they're using the fact that these spores tend to blow on the wind, and then they can actually predict if they find disease in a particular location where it's likely to blow to next. And so then they can work out the threat that it might pose to particular areas. They can then mobilize, for instance, fungicides, so chemical treatments, to make sure that they're protecting the crops in the best possible way. And so this was a huge effort to build all of this infrastructure, human capacity, all of the knowledge, and working as well with the farmers in the region. But there was one key component that was missing, and that was the ability to define the precise strain that was present in a farmer's field. So they could go out to the farmer's field. I mean, you can see it when you look at a field. The field turns yellow when you have yellow rust. And you knew that you had rust in that area. But when you saw it in the field, you don't know what threat it's going to pose. You know it's there, but you don't know the strain. So you don't know which wheat varieties are going to be vulnerable. And so it's really critical that we have strain-level diagnostics. And this is also the case with human disease. If you get a disease and you don't know which strain you have, you don't know the most appropriate control measures. And so it's really important that we have strain-level diagnostics, but also that this works in near real time. So we know exactly which strains are present right now, not what was present 6 months ago, but what's present right now in the country. And so that's where we then decided to step in and try to help by implementing new techniques to try to address this missing component. Okay, so how do we rapidly identify new strains? And here I firstly wanted to introduce you to my collaborator, Dave Hodson at CIBIT. So he works at the International Maize and Wheat Improvement Center, and they're based all over the globe, but their headquarters is in Mexico. And so the work I'm going to talk about is very much a collaboration between my lab and Dave's team, and also with all of the local scientists on the ground. But I also wanted to introduce you briefly to somebody else. And this is a fictional detective known as Miss Marple. So some of you might know Miss Marple, your parents might definitely know Miss Marple, but the method that we developed is named after this fictional detective, Miss Marple. And so we developed this method, which we call MARPLE diagnostics, which stands for Mobile and Real-Time Plant Disease Diagnostics. And so the way that this works is, again, we go out to the field, we get our samples from the field. These are just pieces of leaf or stems. We bring them back to where we're working. And now, instead of extracting RNA, and some of you may know that RNA is quite unstable, instead we extract DNA, and then we focus on diagnostics. And so what we've found is that if we look at many, many hundreds of strains of our yellow rust fungus, if we look at these strains, what we find is that if we look at the different genes, there are some genes that are very variable in sequence, and there are some genes that are not so variable in sequence. And so we can use those that have a lot of variation, and we can take just those genes, and we can look at that variation and work out which strain we have. So we only need a small set of very variable genes in order to work out which strain we have. And so here we do what we call PCR. So what we're doing is we're taking the DNA we've extracted from this infected leaf, and we're pulling out the pieces of DNA that relate to those genes that are very, very variable in sequence. And then we use a portable sequencing device. So this is smaller than your mobile phone, and we can then plug that into a computer anywhere, and we can then do our sequencing. So no longer are we reliant on those really big immobile sequencing machines. We're using a portable sequencing device. And so this is called a MinION, and it's created by Oxford Nanopore, which is based here in the UK. And so then we do all our sequencing, this magical process of sequencing, on a portable device. We then have an automated data analysis pipeline, which allows us to work out which strain we have within the sample we took from the farmer's field. And that information will go directly into the early warning system. And so what's really neat about this method is not only is it portable, it's keeping it in the hands of the researchers directly in country, it's also very, very quick. So we can go from getting a sample from a farmer's field to knowing which strain we have in just 2 days of collecting those samples. And also what's really nice is the fact that it does stay in country. If we compare it to methods that were being used previously, it was wholly reliant on sending samples to centralized diagnostic labs based in Europe or in the US. And so the scientists and workers in Ethiopia would send those samples overseas. They lost control of those samples. Those samples would then be analyzed by somebody unrelated, and then they would get the information back many months later in many cases. Whereas now they keep control of these samples. They're the ones who have the ownership of these samples, and they're able to process them in just 2 days. And also what was important to us through this process was also that it didn't require access to any sophisticated laboratory equipment. And so everything you need to conduct MARPLE diagnostics is contained in a single hard case that we provide to people. And this creates a truly mobile and rapid response laboratory. So if we suspect something's going on in the field, we can move this to wherever we're working, and we can do strain level diagnostics. And so what are the advantages of using this MARPLE diagnostics method? Well, we get information, as I mentioned, in near real time. We can then put that into our disease forecasting. So if we know we've got a new strain in an area, we can then predict where it might move next. It will give us an early indication of new strains that might enter a particular area, such as in the case of Ethiopia and East Africa. And this can help us also with selecting the most appropriate wheat varieties to recommend to farmers to grow, because we can then know that which particular wheat varieties might be vulnerable to those particular strains in country. And also, through this method, we can also monitor other genes in the fungus, and some of the genes that we look at are those which encode proteins which are targeted by fungicides, so chemicals that are used to treat disease. And sometimes, unfortunately, those gene sequences change. The proteins are no longer inhibited by these fungicides, and so those fungicides are no longer effective. And so we can check and make sure that those changes aren't occurring, or at least identify them quickly. And then that allows us to make sure that we're using the most appropriate chemical control as well at the right time for the right strains. And so we've been working with this method in Ethiopia since around 2018 and 19. We've done a lot of trials. We've shown that you can use this method even in the back of a car. So here we had a very simple DNA extraction method that doesn't require cold chain storage. It's completely independent of access to the internet. You don't need any stable electricity. Everything we do is run off battery packs. And all of the analysis is fully automated, so totally removing that need for access to high-level expertise. And even in the back of the car, when I ran this in the back of the car, we were able to show that within 2 days you could take your sample from the field and know which strain you have. So that whole process was done in just 2 days. And so that information then goes out to stakeholders to inform them about what strains are present in countries. So these would be, for instance, farmers or agronomists in the area. And since we started this program back in 2018, we've also trained other scientists. And recently, over the past year or so, we've moved to other locations. And we're also running this method in Nepal and Kenya. And we've trained over 100 scientists directly in these countries, working with scientists on the ground to train them in this method. And it's very, very simple. And they're all now running MARPLE hubs in these areas. And so we have a number of locations that are running this kind of high-level genomic diagnostic technique. And we're particularly working in Ethiopia and Kenya and also in Nepal. So we've been supporting people there. And over the next 12 months, we will move and we will start also supporting new scientists in Tanzania and in Pakistan. And then also we've been working to support other people to adopt this method in many countries, which are shown in blue here. Across the world. So they've also been adopting MARPLE diagnostics, and it's now becoming a method that people really go to for genotyping rapidly exactly which strains of the wheat rust they have in their fields. But we don't know where all the locations are where MARPLE is being run, because also we made sure that everything you need to run MARPLE diagnostics is publicly available. So there's a full training guide through to the equipment list, through to the code, all present on this one website. So there are many other locations, I'm sure, that are running MARPLE diagnostics that we're not aware of, and that's really great to see this being adopted so widely. Okay, so then in summary today, I think we've looked at how these wheat rusts are notorious diseases of wheat that have threatened wheat production since really the dawn of agriculture, and they continue sadly to threaten wheat production today. And we've been looking at how we can actually use this field pathogenomics, so all of the advances in sequencing technologies, the phenomenal new approaches that are coming out, and how we can use these kind of methods for tracking individual strains of the wheat rusts, but also on the plant side, looking at how we can use all of these revolutions in genomics to identify new sources of resistance. And then finally, also looking at how we can implement these new exciting methodologies such as MARPLE diagnostics, which is a genomic-based approach that is really revolutionizing diagnostics, particularly for resource-poor areas. So how we can take these methods and make them accessible to the widest possible audience. And so then I'd just like to finish by, of course, thanking everybody who actually does the work that I have the privilege of presenting. So this is my team here. And I'd particularly like to acknowledge Pilar, who was the one who worked on the analysis of all the genes in the plant that are required to support pathogen infection. And this work is now being continued in the lab by Sarah Bailey, who's also here today and will be leading one of the tutorials. And then I'd of course like to acknowledge all of our funders and many, many, many collaborators around the world. And if you're interested in these various different approaches and methods, then there are a number of papers and our website that I can recommend that you go to read as well. So thank you very much for your attention.